Your mitochondria are not just producing energy, they are producing peptides, signaling molecules, hormones, and they have been doing it your entire life. One of them was only discovered in 2015. It is encoded in the mitochondrial genome, not the nuclear genome that contains most of your DNA, the mitochondrial genome, the separate ancient bacterial origin genome inside every one of your cells. It is called MOTS-c, mitochondrial open reading frame of the 12 srRNA type C. It is 16 amino acids long. It circulates in your blood. Its levels are high when you are young and metabolically healthy. They decline with age, with obesity, with insulin resistance, and with sedentary behavior. When researchers injected it into old obese mice, the mice lost significant weight, regained insulin sensitivity, and their physical capacity improved dramatically. Aged mice on MOTS-c ran further, ran faster, and lived longer than untreated controls. Here's the thing that makes MOTS-c genuinely unusual in this space. It is not a synthetic compound designed in a laboratory. It is a peptide your own body produces, a signaling molecule from your own mitochondrial genome. The question researchers are trying to answer is not whether it works. The preclinical data is compelling. The question is whether supplementing it exogenously does what the endogenous molecule does. WADA, the World Anti-Doping Agency, added MOTS-c to the prohibited list in 2024. Not because it has been confirmed to work in humans, because the preclinical data is strong enough that they consider it a performance-enhancing risk. That is the state of the evidence. Compelling animal data, no completed human trials, banned in sport, widely used in the biohacking community. This episode is the complete picture. To understand MOTS-c, you need to understand where it comes from. And the origin story is genuinely one of the more remarkable things in modern biology. Every cell in your body contains mitochondria, hundreds of them, sometimes thousands, depending on the cell type. Muscle cells are packed with them, neurons are packed with them. Fat cells contain far fewer. Mitochondria have their own DNA. This is because mitochondria were originally free-living bacteria, specifically alpha proteobacteria that were engulfed by an ancestral eukaryotic cell approximately 1.5 to 2 billion years ago. Instead of being digested, a symbiotic relationship formed. The bacterium became the cell's energy factory. Most of its genes migrated to the host cell's nuclear DNA over evolutionary time, but not all of them. The mitochondrial genome retained 37 genes, 13 protein coding, 22 transfer RNAs, and 2 ribosomal RNAs. For most of the 20th century, scientists believed that was the complete inventory of the mitochondrial genome. In 2001, a peptide called humanin was discovered, encoded in the 16 srRNA region of the mitochondrial genome. A short, open reading frame that nobody had been looking for because the prevailing assumption was that the rRNA regions were non-coding. That assumption turned out to be wrong. In 2015, a research group led by Pinchas Cohen at the University of Southern California discovered MOTS-c, encoded in the 12 srRNA region. Another short open reading frame in a region that had been considered non-coding for decades. MOTS-c is 16 amino acids. The sequence is met arg trp gln glu met gly tir ile fe tir pro arg lis loy arg. It is synthesized in the mitochondria, but unlike most mitochondrial products, it can move. It exits the mitochondria, enters the cytoplasm, and during metabolic stress, exercise, caloric restriction, hypoxia, it translocates to the nucleus, where it directly regulates gene expression. A peptide from the mitochondrial genome going into the nucleus to control nuclear genes, mitochondrial nuclear communication. And this is a signaling axis that biology textbooks did not contain until very recently. MOTS-c's primary mechanism of action runs through AMPK, AMP activated protein kinase. This is worth spending time on because AMPK is one of the most important enzymes in metabolic biology. AMPK is a cellular energy sensor. It monitors the ratio of AMP to ATP inside the cell. When energy is low, when you are exercising, fasting, or under metabolic stress, AMP rises relative to ATP, and AMPK activates. When AMPK is active, it does several things simultaneously. It promotes glucose uptake into cells. It stimulates fatty acid oxidation, the burning of fat for energy. It inhibits fatty acid synthesis, the production of new fat. It promotes mitochondrial biogenesis, like the creation of new mitochondria, and it inhibits mTOR, one of the primary drivers of cellular growth, and at excessive activity, cellular aging. AMPK activation is essentially what happens to your metabolism when you exercise or restrict calories. It is one of the primary mechanisms behind the health benefits of both. MOTS-c activates AMPK through an indirect but elegant pathway. It inhibits a part of the folate cycle, the metabolic pathway that processes folate into forms the cell uses for DNA synthesis and methylation. Specifically, MOTS-c inhibits the enzyme MTHFD2, which is involved in converting folate intermediates in the mitochondria. This inhibition causes an accumulation of a metabolite called AICAR, 5-amino imidazole 4-carboxamide ribonucleoside. AICAR is a direct activator of AMPK. So MOTS-c leads to folate cycle inhibition, leads to AICAR accumulation, leads to AMPK activation. This is why MOTS-c has been described as an exercise mimetic. It activates the same downstream pathway that exercise activates through a different upstream mechanism. The comparison to metformin, the world's most widely used diabetes drugs, is relevant here. Metformin also activates AMPK, primarily in the liver. MOTS-c activates AMPK, primarily in skeletal muscle. Both activate the same master switch. Different sites of action, potentially complementary. The second mechanism is less understood, but may ultimately be the more important one. Under metabolic stress, MOTS-c translocates from the cytoplasm into the cell nucleus. Once inside the nucleus, it binds to promoter regions containing antioxidant response elements, ARE sequences, and upregulates the expression of stress adaptation genes. The NRF2 pathway is the most significant target. NRF2 is a transcription factor, a protein that activates gene expression. It is one of the master regulators of the cellular antioxidant response. When NRF2 is active, it turns on genes that produce glutathione, superoxide dismutase, and other antioxidant enzymes. It reduces oxidative stress at a gene expression level, not just by scavenging free radicals. MOTS-c's nuclear function means it is not just flipping a metabolic switch. It is reprogramming which genes are expressed under stress. This is an epigenetic adjacent function, changing gene expression in response to environmental signals without changing the DNA itself. The practical significance when your cells experience metabolic stress, MOTS-c, is part of the system that adapts to that stress at a genetic level. Declining MOTS-c with age means declining adaptive capacity. The cells become less able to respond to stress appropriately. This is one of the proposed mechanisms linking MOTS-c decline to the accelerating metabolic deterioration seen in aging. The third mechanism is anti-inflammatory, and it has implications that extend well beyond metabolic disease. MOTS-c suppresses the NF-kappa B pathway. NF-kappa B is arguably the most important pro-inflammatory transcription factor in human biology. It drives the production of inflammatory cytokines, TNF alpha, interleukin 6, interleukin 1 beta. These are the molecules that cause the chronic low-grade inflammation associated with obesity, aging, cardiovascular disease, and neurodegeneration. The term for this chronic inflammatory state in aging is inflammaging, okay? Inflammation aging. It is not the acute inflammation of infection or injury. It is a persistent, low-level inflammatory activation that progressively damages tissues over decades. It is one of the most reproducible features of biological aging across species. MOTS-c suppresses NF-kappa B signaling and reduces inflammatory cytokine production. In animal models of sepsis, obesity-related inflammation, and ischemia reperfusion injury, MOTS-c treatment consistently reduces the inflammatory response and improves outcomes. The 2024 study published in Theranostics demonstrated another inflammatory mechanism. MOTS-c facilitates membrane repair by helping transport the protein TRIM72 to damaged cell membranes. This was demonstrated in human participants and in vitro, showing that moderate intensity exercise increases MOTS-c production, and that this is associated with improved membrane integrity in muscle cells. This is potentially relevant for recovery from exercise, for muscular dystrophy research, and for ischemia reperfusion injury in cardiac events. The fourth mechanism ties everything together and explains why MOTS-c is specifically interesting for longevity research rather than just metabolic disease. MOTS-c levels decline with age. This is not a minor or contested observation. It has been replicated across multiple studies in humans and animal models. Circulating MOTS-c in humans is lower in older individuals, lower in individuals with obesity, lower in individuals with type 2 diabetes, and lower in patients with chronic kidney disease. The decline is not uniform. It appears to be partly driven by mitochondrial dysfunction. As mitochondria accumulate damage with age, their capacity to produce MOTS-c declines along with their other functions. This creates a hypothesis. Declining MOTS-c is not just a marker of aging, it may be a driver of it. As MOTS-c falls, AMPK activity falls, NF-kappa B activity rises, nuclear stress response genes are less activated, and the cell's capacity to handle metabolic challenge deteriorates. The result is the metabolic phenotype of aging: insulin resistance, obesity, inflammation, reduced physical capacity. The Japanese centenarian data is striking here. A 2015 study in Aging Cell examined a specific mitochondrial DNA variant, M.13828 to C, located in the MOTS-c encoding region that changes an arginine to a threonine in the peptide sequence. This variant is found almost exclusively in Northeast Asian populations. The study found it was significantly enriched in Japanese centenarians compared to controls. The hypothesis: this variant may produce a form of MOTS-c with slightly different or enhanced activity, contributing to the exceptional longevity observed in Japan. This is a hypothesis, not a confirmed mechanism, but it is the kind of observation that drives serious research forward. The animal data on MOTS-c is the foundation of everything else and deserves a careful examination. The landmark 2015 Cell Metabolism paper by Lee et al. Established the basic biology. In young mice fed a high-fat diet, MOTS-c treatment prevented obesity and improved insulin sensitivity. In older mice already obese, MOTS-c treatment reversed obesity and restored insulin sensitivity. The mechanism was confirmed as AMPK dependent. The effects were blocked when AMPK was inhibited. The 2021 Nature Communications paper, the most important MOTS-c paper to date, examined physical performance in aging. Middle-aged mice treated with MOTS-c showed significant improvements in treadmill performance. Old mice, equivalent to approximately 80-year-old humans, showed dramatic improvements. Treated old mice ran further and faster than untreated age-matched controls. Their skeletal muscle metabolomics showed a pattern resembling younger mice. Lifelong intermittent treatment starting at middle age extended median lifespan. In male mice, the effect was modest but reproducible. Importantly, the mice treated from middle age outperformed those treated only in old age, suggesting that earlier intervention produces better outcomes. The sepsis data is also notable. In multiple models of systemic inflammation, MOTS-c treatment dramatically improves survival rates. This is relevant not just for critical care medicine, but for understanding the peptide's anti-inflammatory potency. Two important caveats apply to all of this animal data. First, the rodent to human translation problem applies here as it does to every peptide in this space. Most compounds that extend lifespan in mice do not translate to humans. Second, the doses used in animal studies are high. The standard mouse dose is 5 to 15 milligrams per kilogram of body weight per day. Translating allometrically to humans suggests equivalent human doses would be very high relative to what is typically used in the biohacking community. Whether lower human doses produce meaningful biological effects is not established. The human data on MOTS-c is limited. This is the honest picture. There are no completed randomized controlled trials of MOTS-c as a therapeutic. The clinical trial NCT 03998514, a MOTS-c analog study for fatty liver and obesity, was registered but has not published results as of mid-2026. What exists in humans falls into two categories: observational data. Multiple studies have measured MOTS-c levels in human populations and found consistent associations. Lower MOTS-c is associated with obesity, type 2 diabetes, insulin resistance, chronic kidney disease, and older age. Higher MOTS-c is associated with exercise, caloric restriction, and healthier metabolic phenotypes. A 2024 study in the European Journal of Pharmacology measured MOTS-c levels in diabetic patients with heart failure treated with SGLT2 inhibitors, a class of diabetes drugs that have proven cardiovascular benefits. MOTS-c levels rose significantly with SGLT2 inhibitor treatment. This suggests that at least part of the cardiovascular benefit of SGLT2 inhibitors may operate through MOTS-c upregulation. That is a significant observation, even though it does not confirm that exogenous MOTS-c is therapeutic. Mechanistic data, the 2024 Theranostics study mentioned in the inflammation section, is one of the few studies to demonstrate a direct MOTS-c effect in human participants, specifically showing that MOTS-c correlates with TRIM72 and membrane integrity in humans exercising. The 2025 Nature Medicine paper from Seoul National University Hospital showed MOTS-c prevents pancreatic islet cell senescence in models relevant to human diabetes. This is not a human trial, but it demonstrates mechanism in human relevant biology. The bottom line, the observational associations are strong and consistent. The mechanistic data is building. The controlled human intervention data is absent. Every clinical claim made in the biohacking community about MOTS-c dosing, effects, and protocols is extrapolated from animal studies and community anecdote. One of the most interesting aspects of MOTS-c biology is its relationship with exercise, and it runs in both directions. Exercise increases circulating MOTS-c. This has been demonstrated in human studies. Moderate intensity aerobic exercise significantly elevates plasma MOTS-c levels. This elevation appears to be one of the mechanisms by which exercise improves insulin sensitivity and metabolic health. MOTS-c is a myokine adjacent signal released by working muscle and mitochondria under stress. And MOTS-c, when administered, mimics exercise effects. The same pathways that exercise activates, AMPK, glucose uptake, fatty acid oxidation, mitochondrial biogenesis, are activated by MOTS-c through the folate A-ICAR pathway. This creates a remarkable picture. Exercise makes your mitochondria produce more MOTS-c, and MOTS-c does some of what exercise does. The molecule is simultaneously an output of physical activity and a mediator of its benefits. The practical implication in young, healthy, exercising individuals, endogenous MOTS-c production is robust. As people age, become sedentary, or develop metabolic dysfunction, MOTS-c production falls. The question the research is trying to answer is whether supplementing MOTS-c exogenously can partially restore the metabolic state associated with higher MOTS-c, either as a supplement to exercise in aging populations or potentially as a therapeutic for people who cannot exercise adequately. WADA added MOTS-c to the prohibited list in 2024 precisely because they assessed this relationship and concluded that exogenous MOTS-c could provide performance advantages to athletes. The assessment was based on animal data and mechanistic understanding, not demonstrated human performance enhancement. But the decision reflects the scientific consensus that the mechanism is credible enough to take seriously. The WADA prohibition of MOTS-c effective 2024 is worth examining carefully because it tells you something important about where the scientific community places this compound. WADA does not ban compounds that are implausible. Its prohibited list reflects scientific assessment of both efficacy and detectability. The fact that MOTS-c was added to Section S2, peptide hormones, growth factors, related substances, and mimetics, alongside established performance enhancers like EPO and growth hormone, indicates that the evidence base is considered credible. The specific WADA language describes MOTS-c as heavily marketed by wellness and anti-aging clinics and on social media as a weight loss peptide, even though it is an experimental peptide not approved for human therapeutic use. This framing captures the tension precisely. It is experimental, it is not approved, and yet the performance enhancement concern is real enough to warrant prohibition. And for the biohacking community, the WADA ban has had limited practical impact. MOTS-c is not approved for human use anywhere, is not a scheduled substance in most jurisdictions, and is available as a research peptide. But the ban adds regulatory context and is relevant for any athlete subject to doping controls. For the scientific community, the ban is an implicit acknowledgement that the mechanism is sound. WADA's scientific committee assessed the animal data, the mechanistic evidence, and the community usage, and concluded the concern was legitimate. 3 questions appear in every community discussion of MOTS-c. Let me answer them directly. Question 1, how does MOTS-c compare to metformin? The comparison is legitimate because both activate AMPK and both have been studied for metabolic disease and longevity. The differences are meaningful. Metformin primarily acts in the liver, inhibiting complex 1 of the mitochondrial electron transport chain, which raises AMP and activates AMPK through a different upstream mechanism than MOTS-c. Metformin has decades of human safety data, established dosing, and proven efficacy in type 2 diabetes. It is approved, cheap, and widely available. MOTS-c primarily acts in skeletal muscle. Its upstream mechanism is the folate AICAR pathway. It has no approved human use, limited safety data, and no established dosing. If you are asking which is better supported by evidence for human use, metformin by a wide margin. If you are asking whether they are mechanistically complementary, yes, they may be. They activate the same downstream switch through different upstream pathways and in different tissues. Question 2. Does exercise make MOTS-c supplementation unnecessary? For a healthy, regularly exercising young adult, possibly. Vigorous aerobic exercise robustly elevates endogenous MOTS-c. If you are training consistently, your mitochondria are being stimulated to produce MOTS-c continuously. The case for exogenous MOTS-c becomes stronger in 3 situations: older age, where endogenous production declines despite exercise; metabolic dysfunction, where mitochondrial capacity is impaired; and physical inability to exercise at sufficient intensity due to illness or injury. Question 3. What is the typical dose being used in the community? Community protocols vary widely, and none are evidence-based in humans. The most commonly reported range is 5 to 10 milligrams per week, administered subcutaneously, either daily or 3 times weekly. Some protocols report higher doses. The animal data used doses of 5 to 15 milligrams per kilogram per day, substantially higher than typical human community use. There is no established safe or effective human dose. Anyone using MOTS-c is operating outside the evidence base. MOTS-c has become one of the more widely discussed peptides in the longevity and biohacking community over the past 2 years. Understanding the appeal is worth a moment. The mechanism is unusually compelling. Unlike compounds that act on a single pathway, MOTS-c touches multiple hallmarks of aging simultaneously: insulin resistance, inflammation, mitochondrial function, physical capacity, and potentially longevity gene expression. The Japanese Centenarian Association gives it a narrative hook that resonates with longevity researchers. The safety profile from animal studies is relatively clean. Thousands of animal studies, no oncogenic signal, no serious organ toxicity at study doses, no hormonal disruption. This is reassuring in the context of a peptide with limited human safety data. The comparison to exercise is psychologically appealing to an aging population that cannot exercise as intensely as they once could. The idea that a compound can activate the metabolic machinery of exercise when the exercise itself is no longer possible or sufficient is genuinely compelling, even if the human evidence does not yet support it. The population using MOTS-c skews older, more educated, and more health conscious than the typical peptide community. These are people who have read the Cell Metabolism paper and the Nature Communications paper. They are not operating on pure anecdote, but they are operating ahead of the evidence and that is a risk that deserves honest acknowledgement. Here is the honest picture on MOTS-c. It is one of the most scientifically compelling peptides in this space. The origin story is extraordinary. A signaling molecule encoded in the ancient bacterial genome inside your cells, produced in response to exercise, declining with age, regulating nuclear gene expression, activating the same metabolic machinery as caloric restriction and physical activity. The animal data is strong, reproducible, multi-study, multi-institution, mechanistically coherent. The human data is almost entirely absent from a controlled trial perspective. Every claim about human efficacy, dosing, and outcomes is extrapolated from animal studies and community reports. The WADA ban is a signal that the mechanism is taken seriously by the scientific community. It is not evidence that it works in humans. The gap between the preclinical story and the clinical evidence is large. The reason it persists is the same reason BPC 157's gap persists. A peptide that cannot be patented has no commercial incentive, driving phase 2 and 3 trials. If you are older, exercise regularly, maintain metabolic health, and sleep well, right? Your endogenous MOTS-c production is likely as good as it can be without exogenous supplementation. Those behaviors are the evidence-based intervention. If you are considering exogenous MOTS-c, you are making a decision based on compelling preclinical data and no human trial evidence. That is a decision only you can make with a physician who understands the evidence base. Subscribe to Peptide Decoded. New episode every Tuesday. Drop a comment. And do you think MOTS-c will ever reach a phase 3 human trial? Given the patent problem, we think it probably will not unless academic research funding steps in. Tell us if you disagree.